Enzymes, natural catalysts expanding the horizons of drug synthesis. WuXi AppTec's Biocatalysis platform leverages their precision and efficiency to accelerate the development of your small molecule, oligonucleotide, and peptide drugs, applying novel enzymes and ultra-high throughput screening to unlock synthetically challenging, high-value products to streamline the process to develop small molecule drugs. Developing enzymes dedicated to producing various novel unnatural amino acids to reduce organic solvent usage and expedite manufacturing of UAAs and peptide drugs. Utilizing precise enzymatic ligation for green and efficient synthesis of long RNAs.
In a late-phase small molecule drug project, we successfully developed an enzymatic process for a key chiral intermediate. The conventional chemical route required 5 steps and afforded only a 40% yield. By engineering a novel enzyme via directed evolution, we simplified the synthesis to a single step and boosted the yield up to 90% during enzyme evolution. We turned the enzyme substrate interactions into mathematical models, which let us pinpoint exactly which regions affected performance. In just 1 month, we completed 2 rounds of directed evolution and increased enzyme activity by over 1,000-fold. Ultimately, we replaced a complex, inefficient chemical system with a highly efficient single enzyme, single cofactor system. This cuts the production time for the chiral intermediate from 3 weeks to 1 and reduced costs by over 70%, dramatically accelerating the drug's path to commercialization.
Synthesizing long-chain oligonucleotides has been a major pain point for our customers. Traditional solid-phase synthesis struggles with poor purity and low yield. We developed highly active and selective ligases by designing ligation sites based on RNA secondary structure. This precise tool acts like molecular stitching and could assemble short fragments into a 150 NT sgRNA, achieving ligation efficiency exceeding 85%. We helped this sgRNA project move faster toward clinical stage.
Drug developers rely on WuXi AppTec for rapid and robust enzyme screening, evolution, fermentation, as well as enzymatic process development and scale-up. We develop optimized solutions tailored to the specific requirements of projects from preclinical through commercial stages. Our industry-leading enzyme library, characterized by high quality and extensive diversity, facilitates rapid screening. Our high-throughput enzyme evolution labs combine enzyme rational design and high-throughput screening, accelerating the timeline to identify a highly selective and efficient enzyme specific for each project. Our fermentation plants are equipped with reactors of various scales. We provide flexibility to rapidly deliver diverse fermentation products, from kilograms to metric tons, to meet the demands of late-phase and commercial projects. Utilizing advanced digital systems, we monitor critical parameters in real time to minimize batch-to-batch variations. Our Biocatalysis platform is now empowering clients to accelerate the development and manufacturing of drugs across multiple modalities. Every enzymatic reaction propels projects forward, driving critical milestones to bring therapies to patients. Move fast. Move forward.